system, and some of the results today in the context we're also thinking about in a changing ecosystem. What might we see tomorrow? How do we best prepare for these types of emerging species and prospective industries that could come from that? So we'll see an example of trying to prepare for that in the case of blue crab and getting prepared for that. So hope you enjoy today's meeting. Again, benthic ecology seemed, given the extensive work that us as a research community have been putting towards this effort. And with that, I'll leave it to our presenters to dive in.
All right, thanks everyone. Just bear with me as I get the presentations going. All right, so our first speaker is going to be Shana Harkins, talking about the response of benthic communities in Narragansett Bay to changing temperature and nitrogen input.
Thanks, Grant. Hi, everyone. Welcome to the RIMFI conference. Thank you for coming. I'm going to kick off our talks with the response of benthic communities in Narragansett Bay to changing temperature and nitrogen input. So, okay. There we go. Okay. So just as a little bit of background, there was a successful mitigation in the decrease in wastewater treatment facility nitrogen input to the bay between 2005 and 2013. And that was due to the natural weak summer stratification that the bay goes through every year. And combining that with what peaked in 1980 of eutrophication from the nitrogen input, as well as a strong spring neap tide. Combining all of those elements, you can actually create what I would call the perfect storm for anoxic events. And that actually did happen in 2003 with the Greenwich Bay fish die-off that was experienced. And so as a result of that, there was a mandated decrease in the nitrogen input that was successfully completed. Despite that, there is continued increase in development throughout the bay, and there is continued warming due to climate change. We also know that monitoring benthic organisms actually does gauge the overall health of Narragansett Bay because they are at the threshold where anoxia and hypoxia begin at the sediment water interface. We also know that there is a wealth of historical data, and this table actually shows you how far back it goes. What I bolded are the diver collected core method studies that were done, and they are actually directly comparable to the study that was done currently because we also implemented diver collected cores. The other ones are done by clamshell buckets and grabs. They're a little more robust. They don't happen to keep the water sediment interface that has a lot of flocculent sediment tubes. So they're not as directly comparable, although you can do presence absence comparisons on them as well. But what you can see is that there are comparable studies historically in both Providence River and North Jamestown. Also, the historical studies don't necessarily consider epifauna, which are the organisms that live on top of the sediment. Most consider macrofauna, which live inside the sediment. We did both. And then they also don't consider biomass. Most of these studies did abundance counts of each species, but didn't consider biomass. And so the objectives of the current study are to compare current macrofaunal community composition in a north, mid, and south station in the West Passage of Narragansett Bay, which represents the compare the current between the same station. So adding an increased size range to what we're considering to be the benthic community within the bay. We also want to compare the current north and mid station macrofaunal community, which is the organisms that live in the sediment, to the historical data that we've compiled against nitrogen and temperature data. The nitrogen and temperature data was pulled from the long-term plankton time series because it matches up with our mid station and it matches up with the years that we are studying historically. And lastly, we want to obtain biomass and production measurements of current macrofaunal and epifaunal communities at each station to implement into Narragansett Bay food web models. And so for the field methods, we had, again, 3 stations throughout the bay that represent the anthropogenic influence gradient of the bay. The first one was near Connecticut Point up in Providence River. The second one was in north of Jamestown. It's a very well-studied area in the bay. And the last one is at the bottom, Rhode Island Sound. And at each station, we did sampling at the end of June 2021 and 2022, which represents the peak abundance in Narragansett Bay of benthic organisms, and both diver collected cores and beam trawl replicates were done at each station. So 15 replicate sediment cores were taken, 2 replicate 15-minute beam trawl tows were towed for the epifauna. All samples were stored in 4% buffered formaldehyde and dyed pink to ease identification, and physical data was taken at each station. The lab methods, samples were sieved through a 500 micrometer mesh, which is the lowest size range for macrofauna. Organisms were identified down to the lowest possible taxonomic group. They were long-term stored in 70% ethanol, and biomass measurements, large epifauna were wet weighed on the boat. Wet weights were pooled on a gram scale for larger organisms, and then for smaller organisms, a size range was taken for individual masses on a microgram scale. For statistical analysis, what I bolded is what I'm currently going over, and what's not bolded is everything else. So for the current study, macrofauna and epifauna, Bray-Curtis similarity matrix was rendered, and this was all done in Primer, which is a statistical analysis program done for community statistics. Multidimensional scaling plots were rendered for macrofauna and epifauna, and abundance biomass comparison curves were rendered for both epifauna and macrofauna at each station and in each year. For macrofauna comparison through time, the same thing was done, but on top of that, we also did an analysis of similarities to show how significant the changes were through time for the groups. So how similar are each community through time? And then similarity percentages actually show which species are causing the significant changes through time for the communities. Lastly, a bio-NV plus BV step or best analysis was done to actually see which environmental covariates caused the significant change in community composition through time. So for results that I am currently going over today, this is what is called a macrofaunal multi-dimensional scale. I want to say right off the bat that the dimensions of the axes actually don't have value. What it's showing is the similarity between the replicates for each station and each year rendered visually on a plot. So the more similar they are, the better they cluster together. So what you can see is that Rhode Island Sound and North Jamestown both cluster fairly well together. And you can also see that there is a clear gradient between the 3 stations. So there is a clear gradient throughout the bay. But what you do see is that in Providence River 2021 versus 2022, there's a clear difference between the 2 years. So there is still year-to-year shifts that occur in Providence River community. These are macrofaunal abundance biomass comparison curves. So what you're seeing is a comparison of the percent cumulative dominance of biomass versus the percent cumulative dominance of abundance for each species. And when it comes to abundance biomass comparison curves, what you're looking for is where the biomass curves lie, where the biomass curve lies relative to the abundance curve. If the biomass curve lies above the abundance curve for the entirety of the plot, then the community is considered undisturbed, because what we look for in macrofaunal communities is biomass is higher than abundance. So you have large organisms that are bioturbating lower, and they are longer lived, whereas disturbed communities tend to have higher abundance and lower biomass, with a lot of small organisms hanging out at the top and not bioturbating down very far. And what you can see is that throughout the bay, for both years, the communities are undisturbed and considered healthy. And this is just the MDS plot of the beam trawl replicates. And what you can see is a similar outcome from the macrofaunal MDS. And there's actually a gradient. It's not as well plotted, but there are also less replicates in the beam trawl study. And this is just the ABC curves for the epifaunal communities. And again, there is biomass above abundance for the entirety of the plots for all stations in all years throughout the bay. So again, undisturbed communities throughout the bay. And then for the through time comparison, so there's a lot going on here, but I want to point out that in red, the red boxes are showing Providence River, and you can actually see that it starts over here for the 1970s, but then it flips over to the other side, and then it settles somewhere in the middle for the 2022 data. And similarly with North Jamestown, there's a cluster of 1970s and 1980s data in the middle, and then it sort of shifts diagonally. And again, it doesn't really matter where it shifts, it's just the fact that it does shift in the 2000s. And then again, settles somewhere in the middle for the 2020s. So there is a clear shift in community composition through time. But due to the, according to the best analysis, it's not significantly changing with yearly temperature or yearly nitrogen. So rho value from a best analysis is similar to a R statistic correlation in that if it's near zero, it's considered non-significant. And what I'm showing above are actually just plots of directly averaged dissolved inorganic nitrogen and dissolved and bottom temperature against year. And what you can see is that nitrogen decreases non-significantly and temperature increases non-significantly. And again, these actually match up with the non-significant result of both yearly temperature and yearly nitrogen. So if they're not changing significantly with time, odds are they're not going to change significantly with community composition through time either. So, and then this finally is just the rendered total biomass and total energy in joules and kilojoules for each station and each year for both macrofauna and epifauna that can be put into Narragansett Bay food web models. And so for conclusions, we've found that in the upper bay, there is a community composition shift year to year that still exists for macrofauna. And I say still because this was actually also found in the previous study done by Calabretta and Oviatt in 2008 that found that upper bay stations tend to have opportunistically driven year to year shifts. But we also saw that the abundance biomass comparison curves didn't have disturbed communities. So the upper bay seems to be opportunistically driven, but is still considered undisturbed. And again, biomass measurements were obtained that can be put into Narragansett Bay food web models. But we still didn't see a significant change with nitrogen or temperature through time with the macrofaunal community. This is still an outcome, a successful outcome to mitigation because total nitrogen was decreased, and there could be indirect effects that we're seeing. For example, chlorophyll abundance might change more significantly with macrofaunal community because it's sort of an interim with nitrogen and temperature in the macrofaunal community. It's also possible because we know and we saw that the community changed through time. What caused it? Well, it's also possible that heavy metals and hydrocarbons, which were also decreased on about the same timeline as nitrogen, may have caused this change in the community. It's also possible that what makes up total nitrogen, ammonium or nitrate alone, may also be causing the significant change. It's also lastly possible that analyst bias may come in. So different identifiers are identifying within the study. So it's a different person identifying each organism each time. So moving it up a little more robustly to say functional feeding groups or at a higher taxon level rather than just identifying down to the species may help curb the analysis bias and maybe show a more significant effect. And with that, I'd like to thank everyone that was involved because science cannot be accomplished alone. Thank you, everyone, and I'll take your questions. Please.
Did you, what kind of oxygen data did you use? Because you're going out 15 times the sample, it's only 15 measurements. Did you use any of the long-term data sets to look at like between 21 and 22, that kind of thing at all?
Do you mean, I'm sorry, could you elaborate a little more?
Oh, the continuous oxygen measurements that Candace does at the buoys, are you using them at all to look at changes in terms of the environmental and oxygen changes per year?
So oxygen doesn't necessarily come into play here, mostly because we're not sure at what timeline benthic organisms react to changes in oxygen, and it is constantly shifting. No, we did not use oxygen data.
I would suggest you might want to look at that, because maybe like 21 was a bad year
Yes,
and 22 was a good year.
Right.
Because I mean, you saw those radical changes.
Yeah.
You're not seeing significant response to the other stuff,
mhm,
but oxygen can radically change the whole benthic community. in
fairly
2
quickly
weeks,
too. Yeah,
or
exactly.
less. So,
Yeah, and so that's been the main caveat with testing against oxygen
Yeah,
is
right.
at what response rate are they actually,
So we
responding to
have,
oxygen?
the value we have now in our monitoring is that since about 99 or 2001, we do have long-term, high-resolution data sets. I mean, you don't
Okay.
have them for 58.
Right, but it could still be comparable between the 2008 study and the current study to at
Right.
least see. No, that's, thank you very much. That's a
Yeah,
very
I
good
just
point.
think it might be interesting to take a look at.
Absolutely, thank you.
In your conclusions, you said some changes might be because of changes in ammonium and nitrate. You wanna
Yes.
elaborate on that?
Yeah, absolutely. So what I am currently testing, the current variable that I'm using for nitrogen is total nitrogen. And I noticed that when I split nitrate and ammonia, and total nitrate is made up of, total nitrogen is made up of nitrate, ammonium, and nitrite. And now ammonium and nitrate, when I split them, they had different trends where ammonium seemed to decrease. It was ammonium seemed to increase and nitrate seemed to decrease in concentration through time. So I wasn't sure if there was a significant change with one directly, if nitrate is more of a direct influence on how much chlorophyll is falling to the bottom, and so clearly compared to especially ammonium. And my other thought was, well, maybe ammonium is toxic to benthic organisms. So if there's an increase in ammonium, it's possible that that might be changing the community composition. But I don't think it's nitrogen. That was just more of an idea, because like I said, they seem to have different trends. So I wanted to test them separately to see if maybe ammonium separately from nitrate rather than just total nitrogen had an effect, if that makes sense.
And are these from samples that were taken when you were doing the benthic sample?
So we have, so what we did is we took subsamples, and I actually just got that data, and we were sent them off to have sediment analysis of carbon and nitrogen content done at BU. It just wasn't done in time for me to put it in this presentation. But what we want to do is test community composition of the current data against the sediment, total nitrogen and total carbon that we got from the current study, because that's obviously a much more direct comparison. It's just we didn't have the data yet. So
ammonia toxicity is highly sensitive to pH, in terms of environments. This is really understood extremely well by the aquaculture community because waste product.
right.
So if you go down that pathway,
Mhm.
I would suggest at least monitoring some sediment pH on that. There are available tables on toxicities, LC50 levels and everything. It's all extremely well looked at.
Cool, thank you very much. My other thought was actually just doing a direct comparison with pH, but I didn't see a long enough, timeline to do it. But I think that, again, that could be something that could be comparable, at least between the 2008 study and the current study, because there is pH data that's available through the 2000s. So thank you, that's a good point, thank you.
All right, thanks. So Shana, I just have one clarification or question from the
Please.
chat. So Stephen Hale was asking the p-value equals 0.037 for macrofauna versus temperature over time. I don't know, Stephen, if you want to elaborate on that.
Oh, I think it was the opposite. Hold on 1 second. I might have mixed them up. Yeah, so yearly temperature was negative 0.121 and the nitrogen was 0.037.
Okay, Stephen, I don't know if you can hear if that answers your question, but feel free to elaborate if you need to. All right, thanks very much, Shana.
Yeah, absolutely.
Appreciate it.
Thanks, everyone.
Okay, so next up we'll have Mike Potter. And he'll be talking about hard clam response to a warming Narragansett Bay.
All right. Hello, everybody. Thank you for having me. I'm excited to talk about the last couple years of research I've been working on, and it's with the hard clam response to a changing Narragansett Bay. Okay, so there were two objectives for this study. The first one, the primary one, was to investigate how the winter-spring phytoplankton bloom affects the hard clam. And there was a secondary objective, which was to investigate the impact of the sediment that the clams were in on their biology as well. So the hypothesis here was that a weakening or loss of the winter-spring phytoplankton bloom would result in diminished recruitment, condition, and growth of the hard clam. And this was based on a variety of factors, namely that the winter-spring bloom has been shown to be a benthic food store for invertebrates, and that the bay is expected to warm 3 to 6 degrees by the turn of the century, and that food and temperature in the literature have shown to be the primary factors controlling the success of the species. All right, so just a little background, at least on my understanding of the winter-spring bloom. I have this graph here for you guys, and it's to look at the connection between the bloom and temperature. So on the y-axis, we've got chlorophyll in micrograms per liter, which is often a proxy for primary production within the water column. That's in the green line. And then we have temperature over time in the red line. This is taken from Conimicut Point, which is very close to our research site where we got our sediment, which was just a little north of that, between Conimicut Point and Green Island in the Providence River estuary. So I want to draw your attention to right around here in January. So the threshold generally for the winter spring bloom is around 3 degrees C for a good sized bloom. So when the temperature gets below that, we see suppression, or the literature supports suppression of zooplankton grazing, microbes that break down and decompose organic matter are also kind of chilling out, and filter feeders are largely dormant. During this time, phytoplankton are able to reproduce to very large numbers in the water column, up to usually 10 to 20 micrograms per liter is what we see in a good sized bloom. And this persists as long as temperatures are cold and nutrients are available. So the general trend here is that it continues to be high until around March, when the temperatures start coming back up, and then we see chlorophyll drop down as these grazers, filter feeders, and the microbial community then become active, and they're actively controlling the phytoplankton growth in the bay. And then for our sediment, so our secondary piece, we chose 2 sites in Narragansett Bay. In this graph here on the right, I've got them both highlighted with an arrow and a star. The first one is in the Providence River estuary, which is known to have a history of pollution from heavy metals, hydrocarbons, things like that. A lot of these things like to stay trapped in the sediment, not so much coming in the water these days. So we want to know if these legacy contaminants were a factor on growth, reproduction, and condition of these animals. So our first year experiment was in that top star in the Providence River estuary, and our second year experiment was in a lower to mid-bay site just north of Jamestown in the historic sediment recovery site. So that second star there. So for our study, we used mesocosms. Mesocosms were new to me when I came to GSO. I'd never heard of them before. But what they are is they're effectively a physical model that we use to emulate conditions in the natural environment while being able to isolate and change specific factors. In this experiment, we were specifically looking at temperature and really it was temperature, but in an offhanded way, nutrients as well. So this is on the leftmost photo is a picture of our mesocosms. We used 9 of them, and there were triplicates for each of our temperature treatments, which we'll talk about in the next slide. The middle picture is a quick schematic. We have a big sediment graph from the Providence River estuary, which sets up our benthos, water coming in from the GSO dock as raw seawater, a mixer to keep things moving, and a heat exchanger in order to control temperature. And the rightmost photo there shows a look from the top down into the mesocosm. The big square in the bottom is our large sediment graph, and at the bottom of the orange ropes here, you'll see our sediment trays, which house our clams, and each one of those trays had 40 clams with a total of about 360 for each year of experiment. So a pretty large number. And then, well, we'll move on to the next one. So we'll take a look at our temperatures. Here we have 2021 experiment in the Providence River estuary for the most part design, and then for our mid-bay in 2022. For the most part, we were able to keep these 3 temperature treatments of a Providence River ambient temperature throughout the year. That's this light blue line. That's our ambient treatment, our cold treatment, which was 3 degrees C cooler, and then our warm treatment, which is 3 degrees C warmer. Now, in the beginning of our experimental year, we had some failures in our system. It's a relatively new system. So instead of starting in January like we'd like, we ended up starting in March. And we used nutrient simulated phytoplankton blooms in lieu of this temperature control. So we had in our ambient treatments, while all the temperatures were the same, we added 8 times the Narragansett Bay average nutrients, which is consistent with the Providence River effluent coming in. We doubled that for our cold treatment, and we added no nutrients for our warm treatment. And we continued to step these up in that regime until we saw the bloom dynamics we would expect at different temperature years. So we would see average grazing in the ambient year, where we see a reasonably good sized bloom, almost no grazing in the cold treatment, so we see a really big bloom, and in our warm treatment, we expect to see no bloom to emulate really heavy grazing on the phytoplankton. So we continued this temperature trend through both years of the experiment. And the second year was almost entirely the same with a few modifications. Now we move on to primary production. We were measuring this by fluorescence within the water column. Once again, we have the first year experiment here on the left and the second year experiment on the right. We're going to focus mostly on this one. We have chlorophyll on the y-axis, and then the months of the year through the experiment on the x-axis. The color scheme is the same. Light blue is ambient, dark blue is the cold treatment, and red is the warm treatment. This is the time where we were adding nutrients, and we saw exactly what we expected to see with the phytoplankton blooms. We saw a good sized average bloom in the ambients, an outrageously large bloom in our cold treatments, and nothing in our warm treatments. And then after we got these blooms established for about 2 weeks, we tapered off. You can see the chlorophyll numbers dropping, and then we just added a supplementary amount of nutrients around 8x per day, consistent with the research site, to carry the experiment through. In the second year, the data looks a little bit different. That system was dominated by benthic macrofauna and benthic diatoms. So we really couldn't measure all of the primary production just by measuring what was in the water column, because most of it wasn't there. But we can see that each of the treatments pretty much had one big bloom spike. All right, so that's all of the background information. Now we want to look at the clams. Remember, we were looking at our recruitment or our reproductive potential of these clams, the condition or health of the clams, and their growth. So a couple different methods here. So looking at the reproductive potential of the clams, we established 3 reproductive stages by looking at them. The first one is engorged. This area here is the visceral mass of the clams where its main body is. In an engorged clam, this is big, so engorged, and it's mostly filled with reproductive material. That's this creamy color here. So an engorged clam was almost entirely dominated by this reproductive material. Partially engorged clam is still a little engorged, but noticeably smaller, and the reproductive material is 50% or less visually. And our reduced, you can see the visceral mass is almost non-existent compared to this guy. These are all similarly sized clams, and there's no visible reproductive material. It's mostly clear and thin throughout. And then for our condition or our health metric, we used a condition index, which was a gravimetric method of looking at how much space the clam's body occupied in the available volume that it has inside its shell. And then for our growth, we measured these clams in a lot of different ways, but for the growth specifically, we looked at a stationary space here, or a spot here on the umbo, where the oldest growth is, to the addition of new rings to determine our growth. All right, and to look at all this data, I looked at it all individually, but I wanted to get a big picture of how they all looked together. So I used an ordered logistic regression model in R, which looked at the relationships between condition, growth, and the treatment the clams were put under on their reproductive potential or their gonad stages. And here are the results. These results reflect the bloom effect. Now, the treatments that had the largest bloom had the most food available. That's the biggest factor. It seems to be in this experiment that it was really the food availability, not necessarily the temperature. But they're connected. like with the bloom. So clams that had a higher condition or they were healthier clams were more likely to be highly reproductive. Not too surprising, probably. Something that was a little surprising to me is the data suggested it was a little weak, but it was slightly significant. Clams that had a higher growth were actually in the warmer treatments, which suggested that, and they also were less likely to be reproductive, which suggests that there may be some trade-off between food availability, reproduction, and growth. And looking at our two really opposing treatments here, if they belong to the cold treatment, which once again had most food, the clams were more likely to be highly reproductive. And the clams that belong to the warm treatment where there was less food, you expect less food and more stress probably, the clams were less likely to be reproductive. And I'd like to point out that these clams are super tolerant of temperature and salinities and things like that. That's why I believe it's more the food availability that's what's affecting them. Now, that's all of our primary objective. Now our secondary objective, we wanted to see how the different sediment was impacting these animals. So I have a density plot here on the right, which shows the Providence River Estuary Year 1 experiment in blue, and the Year 2 experiment using the more pristine mid-low bay sediment in the black line. And we looked at the relationship with the condition index because that correlated very well with the reproductive potential. They were pretty great, but this was a little bit easy to work with, if I'm being truly honest. But as it turned out, there was no significant difference between the sample from either year. So comparing all of these clams from Year 1 to Year 2, you see these density lines. Don't pay attention to the y-axis, that just helps you calculate the area under these curves. What you're really interested in is how these lie across each other and where the mean sits. So based on this, it looked like there was really no large impact. There was no significant impact between the first year experiment or the second year experiment with respect to their sediment that they were in on their health, reproduction, or growth. And conclusions from the experiment, what we set out to look at, do the results support this? For recruitment or reproductive potential, yes. Clams that had more food and a stronger winter spring bloom in colder treatments showed greater recruitment potential. They were also healthier, so their condition was better when they had more food in the colder treatments than when they had less food or belonged to the warm treatments. Their growth, I'm going to say no, because it didn't go in line with what we expected to see, but we did see some implications from the data, which give us good insight to what's happening with these animals. And with that, I'd like to leave you with my acknowledgements. Once again, thank you for having me, and I would love to answer any questions you have. Yes.
Yeah, in this vein, one of my former graduate students, Carolina Merrill King, has a good paper on correlation condition index and GoNet.
Yeah, I peeked at it. I wasn't really surprised to see what we saw for sure. Yeah. Yes.
Hey, Mike, thanks for your talk. I was curious, as part of the work, were you interested to see how the results differed by location and sediment? And if you are, did you collect data to see if the sediments truly are different based on metals or detritus?
We did not. That is something, since this was a secondary objective, our primary objective was to investigate the winter spring bloom. We kept everything as consistent as possible between Year 1 and Year 2, but just swapping out the sediment to see if that really had the impact. There is a lot of literature, even more recent studies by Brown in like 2007. I think I read something in 2012, which was just analyzing what was in the sediment. In fact, I snuck some if you ask me question slides. Hold on a second. It's that one. So this is from an analysis that Brown did in 2003 to 2005, and they published the report in 2007. And I just picked a random, heavy metal lead. And where our mid-bay site is, it's actually right here. And where our upper bay site was in the experiment is right here. And the general trend from lead, cadmium, nickel, most of all except for mercury, actually, that one's pretty well everywhere, showed that this right here is the effective range low, which basically means it's not really having an effect on the animals, or it's not a really meaningful effect. And then in this area, this is possible effects where like a certain percentage are kind of being negatively impacted. And here is where you're probably in the bad zone for things that live in the sediment. And you can see that there's this trend in all of this data that shows recently that those heavy metals are still there. They're in the top 10 to 15, 5 to 10 centimeters of sediment. So they also did hydrocarbons, but I didn't see anything in that report. But for the most part, they like to glom on and stick to sediment particles. And if you leave them in the sediment, they're fine. But if they get moved up by bioactivation or physical processes, they can then be, filtered by filter feeders, and then they become problematic. But from what I've been able to read, it seems that as long as they stay put, they're pretty good for the clams. But to answer your question, we didn't analyze the sediment in the experiment, but we were basing this off of a wealth of knowledge that says that there's a gradient of these pollutants that still persists but are largely inert for the most part.
Great, thanks.
Yes.
Like, your low nutrient level, you using nutrient levels from where? From GSO here?
No.
Lower?
So the experiment design, we based it off of the nutrient level for the Providence River estuary, so it was all based there. We did have water coming in at GSO, which should have been about the Bay average, somewhere around pretty low. So we were actually supplementing nutrients in to try and model the Providence River estuary,
Oh,
with the exception of the bloom period that we were trying to get those simulated blooms. We were pounding nutrients to make that happen because we were fighting warmer temperatures and increasing grazing at those times. But for most of the experiment, we were trying to model both years based on the nutrient profile in the Providence River.
Great. Thanks very much, Mike. Appreciate it. All right, so next up we have Declan McCormick, talking about early life history of American lobsters.
Thank you.
Thanks, man. Hi, everyone. So my name is Declan McCormick. In the current set, I'll be talking to you about the early life history of American lobsters in coastal southern New England waters. There we go. So briefly, I just wanted to talk about the history of the southern New England lobster stock. So here is a diagram of abundance in millions of lobsters. It's a time series from 1984. Goes all the way to 2018. And abundance, you can see, is in the blue line. And so in the early to mid 1990s, you can see that the stock was particularly robust. And then come late 1990s, early 2000s, there was a steep reduction in the population. And that red line down the middle is the accepted threshold between a high abundance regime and a low abundance regime. So you can see in 2003, the population crossed over into a low abundance regime and has sort of continued to crash ever since until this goes to 2018. And so this is due to a myriad of different conditions. So it's a combination of fisheries mortality, changes in the environmental conditions, as well as the onset of an epizootic shell disease that occurred in the mid 1990s. Sort of blanketing all of this is just general climate warming in the bay is putting a stressor on these lobsters. So we believe that the southern New England lobster stock is currently experiencing a recruitment failure. So this is a diagram of spawning stock biomass with recruitment, and the recruitment's lagged, so it matches up with the year it was spawned. And then this is a time series. It starts in 1979, and you can follow it through down to 2010. So you can see when the population was, when the abundance was high, you can see that the recruitment was high and there was a significant spawning stock biomass. But then in the early 2000s, you can see recruitment has sort of crashed. And we believe that there's a population bottleneck that's occurring somewhere between when the lobsters are hatched and when they recruit to the population. And somewhere along there, they're being bottlenecked and they're not making it to the recruit stage. So the objective of this project is sort of to quantify that recruitment bottleneck and find out where in the lobster life history potentially it's occurring. We'd also like to assess the abundance and spatial distribution of both larval lobsters and post-settlement lobsters in southern New England, and then compare that data with historical data that's been taken previously, and compare that with climate trends to see if this change is happening over time. So just a quick overview of the lobster life history. So here's a schematic. So the lobsters start down here, and then they're born, and their first stage is their planktonic larval stage. And they have 4 substages, stages 1 through 3, and then these planktonic post larvae over here at the top. And so they spend about 3 to 12 weeks in the planktonic stage, and this is concurrent with the summer months. And then at the end of the summer, the post larvae settle down, they dive down to the bottom, and they find suitable settlement habitat, just cobble, gravel, rocky type habitat, and they seek shelter down there. And then when they can leave the shelter after they're large enough, and they become juveniles. And so we'll be focusing in this study on 2 specific life histories stages. So the first one is the planktonic one, and to sample that, we'll be using Newston net tows. So this is a Newston net here. Basically, we'll go out and drag the net through and see if we caught any larval lobsters. And then the second one are those settling post larvae, or the young of year that have settled. And for that, we'll be using passive benthic collectors, which is basically like a squished lobster trap that's filled with rocks, and that is simulating suitable settlement habitat. We're basically creating a nice little home for a small lobster to come and settle. So these are our sampling sites in southern Rhode Island. It's a little small here, but you can see the benthic collector sites are the blue and purple sites. And so we've got, a good breadth around the bay, and then we're around the sound, and then we're up into the bay a little bit. The red and purple sites are where we're doing our larval Newston net tows. And again, we sort of, we're right at the mouth of the bay and towards out into the sound, and we kind of are covering both passages there. And then obviously the purple is where we're sampling both Newston and benthic. And those are important to see how the larval supply is affecting the settlement indices. So I talked a little bit already about larval sampling, but the sampling occurs between around June 1st to August 1st. We sampled in 2021 and this year in 2022. And we had 6 sites, and at each site we did 2 15-minute net tows. And this was with a 1,000 micron mesh, which is small enough that it will catch larval lobsters, but it's large enough that it lets larger copepods pass through, so we're not filtering through a ton of zooplankton. There's a flow meter in the Newston net, and we use this to calculate the volume of water that we sampled, and then we can standardize the abundance of lobsters based on a specific volume. We also collected environmental data, and then these are some of the lobsters that we found from last year. That's a stage 4 at the top, stage 3, and a stage 1. And then this is a lovely Rhode Island day, Newston towing with the sailboats in the background. So the other one is the benthic sampling. And so these are deployed in May, and they're let sit throughout the entire summer and picked up in September. And this allows them really to ingratiate in the bottom habitat and sort of feel much more natural. We have 10 sites, and at each site we deploy a trawl of 10 traps, so 100 traps in total. And like I said before, they're filled with cobble to simulate this habitat. So this is a schematic of a traditional benthic collector up at the top, which is filled with cobbles, maybe about this big. I'll have a picture you can see in a second. But per the advice of some of the collaborating lobstermen who worked on the project with us, we substituted about 25% of the traps with this, what I'm calling new traps, new collectors, which is using the same size cobble, but also supplementing that with smaller gravel pieces. And the thought is that the really tiny lobsters aren't getting enough protection from predation in the big crevices of the large cobble. And so we tried these out as well. We placed them at a variety of depths throughout the bay and in the sound. And then when we pulled the traps up, we measured everything we could and then took everything back to the lab for analysis. So this was when we filled up all of the traps with rocks, giant pile of rocks. And that's kind of the size you can see. And then once we collect them, we take all those rocks and pitch them over and collect all the stuff we found, measure it, and then we haul them back into shore. So I'm still in the process of going through all of the data. So I'm just going to kind of throw everything I have at you guys and try to put a picture together. So this here is the larval abundance for 2021. On the left-hand side are the stage 1 and 2 lobsters. And on the right-hand side, you can see the stage 3 and 4 lobsters. And then this is over the sample period, and you can see there it's larvae per 1,000 meters cubed, which is consistent with historical studies. And obviously, the first thing you can notice is there's a giant gap between our first and second data points. This was due to boat malfunctions last summer. But what we can see is that stage 1 and 2 abundances are pretty high at the beginning of the year, and then they taper off significantly, come mid to end June. And then, of course, they're molting and turning into the stage 3 and 4s, and those sort of peak in July, like mid-July, and then they're almost all settled by the beginning of August. They're come back down a little bit there. So I'll be adding the 2022 data to this, and then we can sort of see how the abundances change throughout time. But this is just a quick look at it. Jumping to the benthic collectors. So we just talked about the larval lobsters. These are the young of year lobsters. So in all of the collectors, we caught 47 lobsters. 36 of them were in the traditional traps and 11 were in the new. So the total average lobsters per square meter, you can see we're catching about half a lobster per meter squared. And that's consistent with traditional and new, regardless of the trap type, when considering the total number of lobsters. When you take a look at the young of year average, while we're dealing with small sample sizes here, obviously, there's a suggestion that the new trap is actually catching, the new type is catching more young of year than the traditional type. And then, so this is a frequency plot of the size distribution of the lobsters that we caught. You can see we're catching a majority of like 30 to 40 carapace length lobsters. Those are like 2 to 3 year old juveniles. We're not catching a ton of the young of year, and this is the delineation. So we caught not a ton of the small lobsters. So putting this in context, this is the time series for the Rhode Island suction sampling, and their average lobsters per meter squared. So for total lobsters, we caught around 0.44, so pretty consistent with that last blue dot, and 0.02 is also very consistent with what we saw for the young of year. So we're seeing similar trends there. And then taking a quick peek at, this was a study done in 1997 in Rhode Island at some of the same sites. It was longer. It was like a 6-year study, so the sample size is much larger. But you can see they're kind of seeing the complete opposite of what we found, which is where they're getting a lot of young of year, and then it sort of tapers off. And if you can squint, you can almost see the cohorts growing, and we've got good representation from the juveniles, very low representation from the young of year. So it begs the question where they're coming from. This is a look at the climate data associated with the larval toes that we collected. So this is surface and temperature, surface and bottom temperature data for all of the sites. And you can see, obviously, there's a positive trend as the year goes on because the water warms up in the summer. But what's to note here is that black line is at 20 degrees Celsius, and that's the accepted lobster stress threshold. So anything above that level is putting some sort of physiological stress on the lobsters, and that's consistent through all phases, all life stages. And so you can see by the end of the sampling period in August, all of our sites are well above 20 degrees Celsius, some up into like 22.5. And as we all know, water doesn't stop warming August 1st, so that's just going to keep skying up. When looking at the bottom temperatures, what's interesting is at the end you can see that there's a nice gradient, and this is, as you might have guessed, this depth gradient there. So the pink orange sites are our deeper sites, and then the red is our shallowest. So you can see lobster settles on August 1st. They're going from 22 and they dive down and they only get 21.5. So it's still pretty hot down there. Just briefly, for anyone interested in crabs, this is some of the crab data that we found. I'm sure there's a few in here. So this is from 2021 in the benthic collectors. This is the size distribution of Jonah crab, rock crab, and black-fingered mud crab. And you can see that comparatively, we're talking frequencies of 10 to 15 with the Jonahs and rocks, and we're talking like up to 2,000 for the mud crabs. So dominated the benthic collectors. But you can see, we're getting a pretty normal distributed size, right around 30 to 40 millimeter carapace length for both those crabs. And then, so while I'm still picking through the data, so just some future directions to further test the differences between the new and traditional trap collector. I did a two-way chi-squared analysis, and I binned off the lobsters into 10 millimeter increments, up to from 5 millimeters up to 55 millimeters, and then comparing new versus traditional. And there was a significant difference between the new and traditional, albeit we're dealing with, again, small sample sizes here, so be taken with a grain of salt. But there's a large suggestion that the new collectors are catching significantly more young lobsters than expected. And even it's showing here that the traditional traps are catching less than they should be. They're not just catching like a normal amount, they're catching potentially less than what would be expected. So this is something I'm going to investigate further in. And then also interested in potentially, once we get more data in here, fitting a polynomial equation and integrating under the curve so we can get a total abundance for the entire summer, and we can use that to better compare with total abundances that have been seen previously in previous studies. And that is all I have for you guys. And I'd like to thank everyone up here, and I'm happy to answer any questions.
I'm going to demonstrate my glorious lack of understanding of shellfish for this question. So I watched 3 really good talks now, and the one thing that struck me is, and you may collect this, you may just not have presented it, is any information about the geography of where the bottom dwellers are, or even the midwater ones, and then also, do you collect any sort of optical data related to where you're sampling? Because it would seem to me, with the amount of work we can do with super high resolution satellites or even optical instrumentation, there might be a turbidity, water clarity, tracking, or connection to why they're living where they are.
Yeah, no, that's a great question. To answer the first part of your question, we do some of the sites that we picked for the benthic collectors. They were chosen because we know that they're good settlement habitats, even without the collectors. That just naturally, that's a spot where lobsters will settle. And additionally, we've been communicating with the lobster men that help us on this project and picking their brains and seeing where they're finding the smaller lobsters. And so using that as a guideline to decide where to place the traps. As far as like actual bathymetry, I mean, we just have the depths for each site. And then we didn't take any turbidity data, but it's interesting. I think that's a good point to potentially look into something like that.
Or even changes in the optics of the water.
Yeah.
It could tell you a lot about, well, it could give you if you start doing it right and you have the time series, I think it gives you some insight into how you might track the natural habitats and the migration from space, depending on the water depth and clarity. But you have to build a relationship there, and I don't think it would be that hard to do.
Right.
But a lot of the commercial satellites now get down to a centimeter or 2 resolution, which is really incredible.
Yeah.
Yeah, that's great.
Thank you.
Could early settled out lobsters be a food source for some of those crabs that are infecting your benthic traps? And or do you think there could be some sort of like resource competition going on with the abundance of things that you're seeing in those areas too, just from you going out and looking at these things?
Yeah, 100%. As far as predation, I think the little guys are susceptible to predation from a ton of stuff. The mud crabs don't get huge, but they're strong in numbers, I guess. So, but yeah, exactly. As far as competition, yeah, it's something I want to look into, actually being able to test the effect of competition on the prevalence of lobsters. And I just forgot.
Yeah, I wonder if are you taking a snapshot of your benthic traps before you begin to dig through them at all?
We don't typically take a snapshot of the thing before we take it. But with the warming temperatures also, we are seeing higher numbers of known predators of lobsters like black sea bass, cunner. We pulled up one collector this year that had like 55 young of year black sea bass in it. So I mean, those are like young of year eating machines. So yeah.
All right, I do have a question online from Jonathan Spirits. Do you save any of the larvae or post-larvae in ethanol, or is it all in formalin?
It's initially stored in formalin, but then it's put in ethanol. And it is, yeah, we keep it still.
All right, he says thanks.
Yep.
Okay, thanks, Declan. All right, so our final talk will be from Katie Rodrigue, and she'll be talking about assessing blue crabs in Rhode Island state waters.
He's a trooper for dealing with blue crabs. I like to deal with them in the winter.
Yeah, they're really slow.
Just probably the only thing they have. Right. Thank you, everyone. Thank you, Corinne. So my name is Katie Rodrigue, and I'm going to be talking about what is my master's thesis work, which is assessing the blue crab population in Rhode Island state waters. So just a quick presentation overview. So I'll first give you a little bit of background information on the species and its fisheries. I'll talk about what our objective of the study is and why we're doing it. And then the approach of how we're going to look at this, which will have two different parts, and then we'll have some time for some questions. So the blue crab is a portunid swimming crab. It supports recreational and commercial fisheries across the East Coast and in the Gulf of Mexico. It's the highest value commercial fishery in Maryland and Delaware. It's also got a high value fishery in Virginia and New Jersey, and a small commercial fishery in New York. It's a major predator in benthic communities. They're very opportunistic. They'll eat juvenile fish, other crustaceans, shellfish, infaunal invertebrates. And another important aspect about their biology is that when water temperature gets below about 10 degrees Celsius in the wintertime, they will undergo a period of dormancy where they stop growing. They sort of just hunker down under the sediment and wait until it warms up again in the spring. So just to illustrate this, I have a figure here on the right. This is from Maryland winter dredge data in Chesapeake Bay. And they looked at percent mortality as a function of the February bottom temperature. So February being typically the coldest month in the winter. And so you can see in years where there was a warmer winter, they saw pretty low mortality. But as the bottom temperature decreases, the mortality exponentially increases. And a more recent study that also illustrates this. This was in Long Island Sound, Connecticut. This study found a correlation between the number of cold days in the winter, I'll say, compared to the abundance of crabs caught in the following summertime in their survey. So in years where there were a lot of cold days, they didn't catch very many crabs in the following summertime. And this is thought to be the main reason that limits their northern range, and also contributes to their high interannual variability. So their abundances can be very sporadic from year to year. And so just to reiterate, so the objective of the study is to assess the blue crab population in Rhode Island ahead of what could potentially be an emerging commercial fishery. And the biggest reason for why we need to do this is to date, there has been no comprehensive assessment of blue crabs across Rhode Island state waters. But there has been some interest shown in changing regulations to allow for a commercial fishery or an expanded fishery. But of course, in order to make any sort of regulatory changes like that, we need an assessment and long-term monitoring to inform those decisions. There is evidence that blue crabs are undergoing a range expansion and population increase in some areas. There's a study in 2019 in Chesapeake Bay that looked at projected water temperatures up to the year 2100. And their models predicted that winters could be up to 50% shorter in Chesapeake Bay and result in up to a 20% increase in overwinter survival. And this, coupled with a prolonged growing period, could increase blue crab productivity in the future. And then up north in Gulf of Maine, there's also been an increase in observations of blue crabs as well, where sightings tended to be rarer in the past. And there's also some evidence of recruitment occurring in Gulf of Maine where they were not thought to recruit before. And as we all know, Narragansett Bay is also warming. It's particularly warming in the wintertime. So this begs the question, will we see a population increase of blue crabs in Rhode Island as due to climate change? So for the study, we're going to take a two-pronged approach here. The first is going to be a broad analysis of blue crabs across the state from using data from various monitoring surveys. And then the second is the establishment of an annual winter dredge survey to hopefully use as a tool for long-term monitoring. So this is a map of all of the station locations we have for all of the data sets, existing data sets on blue crabs. So we've got 4 beach stain surveys that occur in the summertime, and those are conducted monthly. We have one in Providence River, Narragansett Bay, the South County coastal ponds, and down in Block Island. We've also got a summer fish trap survey that is conducted monthly in Providence River. And then there are 3 trawl surveys that we have blue crab data from. The first is the URI GSO trawl, which is the longest time series that we have on blue crabs. We've got data for them back to 1970, which is great. And then we've also got 2 DEM trawl surveys. So one is a monthly survey that goes out every month throughout the year. And then there's a seasonal survey as well that goes once in the spring and once in the fall. And all of these surveys have a fixed site monitoring design, with the exception of the seasonal trawl, which also has a random component to it. There are 14 sites that are randomly selected each season. And that's the beige dots all over the place. All right, so we're still in the very early stages of this project, but I did start to take just a quick look at preliminary analysis on some of these data sets. So the first is from the GSO trawl. This is percent occurrence of blue crabs by year, so it's not abundance, rather it's the percentage of tows that had blue crabs present in them. So you can see there is quite a bit of variability from year to year, but there's also a clearly increasing trend at both Fox Island and Whale Rock. So blue crabs we're more commonly encountering them in more recent years. And then I did the same analysis for the coastal pond data set. It's worth noting this is a much shorter time series, so we only have blue crab data back to 2009. And you can see there's a lot of variability and not too many clear trends here. And when we combine all of the ponds, you can get a better look at that. And you can see it almost seems like there's like a static percent occurrence there for blue crabs. It's average about 55%. But I think what's so important to note is, we're seeing blue crabs consistently in over half of the samples that we do. And if I don't have the figure here, but if you truncate the GSO data, just to look at the 2009 to present, the average percent occurrence is about 48%. And obviously these data sets aren't directly comparable. They're different habitat types and different size selectivity between the surveys. So I think what this does illustrate is that it seems like the coastal ponds have a fairly high abundance of blue crabs, and they likely serve as very important nursery ground like they do for many other species. So I think it just highlights the importance of us looking at the size structure of the population and how different sizes are distributed, spatially distributed. All right, so next steps for this, really just continue to investigate these spatial and temporal differences in the population. We're also planning to create models that can account for the effects of different abiotic factors such as habitat type, salinity, temperature. We're also going to look into trying to create an overall like basically a single index for blue crabs in Rhode Island from all of these different abundance indices that we have. And that will be, we'll be looking into a hierarchical analysis that was presented by Paul Kahn in 2010. We'll be creating size-based catch curves. So this is going to allow us to track individual cohorts through time, try and look at their mortality rates, also assess seasonal growth, their movement patterns, recruitment, and importantly for management, how do current size limit regulations relate to the actual size structure of the population? And then finally, sex ratio and egg production as a way to estimate spawning stock biomass. And for some of these surveys, we do collect shell condition data so we can look at seasonality of their molting. All right, and moving on to part 2. So this is the establishment of our winter dredge survey. So this is based off of methods that have been used in Chesapeake Bay since 1990. It's considered their most comprehensive and statistically robust survey for monitoring blue crabs, and it's what they rely on primarily for their stock assessments. You can see on the right, that's the dredge that we use. It's a 6-foot Virginia crab dredge, and it's identical to the dredge that they use in Chesapeake Bay. And we tow it from the DEM research vessel Reliance. We use a stratified random sampling design, and each of the strata that we use is different water bodies or regions. We conduct the survey December through March while the crabs are dormant. They're not moving, which is helpful. And we have stations throughout Narragansett Bay and in some of Rhode Island Sound. All right, so we did pilot this survey last winter, try and sort of learn how to do this because none of us had ever worked with this gear type before, so it was quite the learning curve. But we were able to successfully complete 39 tows, and we did catch blue crabs, which was nice. So we caught them in Providence River, Greenwich Bay, a little bit in Mount Hope Bay, and then sort of out front here in Rhode Island Sound. But we didn't see particularly high densities. On the map, the more red squares are where we caught more crabs. So our highest density was about 1 crab per 62 square meters. And that was up at Providence River, one station there, and then also off of Bonnet Shores there to the south. But I think what was exciting, what we saw in this data, is that we saw the spatial segregation that we would expect by sex based on what we know about blue crab biology. So in the summertime, after blue crabs mate, the females move down to the lower parts of the estuary to get ready to release their larvae, while the males and the juveniles will stay in sort of the upper bay and tributary areas, and they'll stay like that for the winter time. And that's exactly what we saw, which was pretty cool. So males and juveniles were exclusively caught in those upper bay sites, while all of the crabs that we caught at those southern sites were mature females. So moving forward, so really just to continue this dredge survey. So we piloted it last year, so weren't at the sampling intensity level we would hope for, obviously. So we'd like to ramp that up for this coming season. Hope to start by January. And I think the main goal for this component is just to determine, is this a useful tool for monitoring blue crabs, or how can we improve it to make it a useful tool? And with that, I'd just like to thank everyone who's been such a huge help on this project and continues to help, and the U.S. Fish and Wildlife Service State Wildlife Grant, which funds the research. Be happy to answer questions.
Great, Tom, thanks. From your map of your sites, it looks like you had 2 sites in Narrow River, which are green.
Yeah, I've got 3 sites in Narrow River. Let's go back there. Yeah, so kind of hard to see because they're like overlapping a bit. So this one up here, the northern part, that's right south of Bridgetown Road. We catch a lot of blue crabs there. That's a lot of freshwater input at that site. And these 2 sites are more at the Middle Bridge area. So don't see quite as many blue crabs there.
But nothing up in the ponds.
No, nothing further north than that. Yeah. So that survey is designed for it. The original intention is to monitor juvenile winter flounder. So it's that's how the sites were selected based on that.
Given all your sampling locations in the coastal salt ponds in Narrow River, I was surprised to see blanks in the Palmer River and Barrington River 100-acre Cove. Are you sampling up there?
Yeah, no, I don't have sampling locations up there. We do. I know Dave Taylor at Roger Williams samples in Seekonk and Taunton River. He's looked at blue crabs quite a bit up there, but we don't have from our survey data. We don't have stations up there.
Believe it or not, South Water Street in Providence, the boardwalk that's there, late spring, early summer, blue crabs
Everywhere. Yeah,
it's amazing the density of blue crabs
that's awesome.
in the Providence River downtown.
Do you see that? Do you think you see that consistently, like every year, or is it kind of?
I can't say.
Yeah, just curious.
consistently, but
Yeah,
when I've looked, they're there.
a lot of them. Yeah, it's very cool. Somebody sent me a video last summer. I think it was Green Hill Pond. So way down by Ninigret there, but just hundreds of blue crabs all leaving the pond at the same time, right under that bridge there. So it was pretty wild to see. And they're there.
Okay, what's the benefit of showing the data in your percent occurrence instead of just total abundance?
So I did do abundance as well, but I didn't show that here today. I was trying to just get an idea, are we seeing them more often really versus, how, but abundance.
But you have those data.
Yeah,
Okay.
absolutely. Yeah, that will absolutely be part of
Very
our
good.
analysis. Yeah, for sure. Yeah.
So correct me if I'm wrong, but I guess you probably didn't get any like egger females in the winter dredge.
No,
So
not in this one.
Is
Yeah.
there gonna be like some sort of other program going forward, or how are you gonna assess reproduction?
Yeah, so I've been trying at least the last couple of years on my coastal pond survey, I have been collecting that data. And I know in our Providence River survey, they've also started to collect that. So we don't have as much of a, like a time series of data, but I'm not sure if that's super important to look at their seasonality anyway. So yeah, we do have some of that data, but not really in the dredge survey.
So are the females moving south towards the coastal ponds to lay their eggs, or do they lay their eggs offshore and then the juveniles settle in the coastal ponds?
I think the latter. So the females move out to more high salinity waters, and then they'll release the larva. It's to allow, I think, the greater dispersal of their larva, which is why they move out like that. Yeah, but it's a good question. Yeah.
And so what you're seeing is the coastal ponds as more of a nursery for juveniles, right?
Yeah, and I mean the survey too is more selective for smaller things, just the nature of that survey.
Right.
But we catch a ton of juveniles, and I do think that it is an important nursery for blue crabs.
Yeah, it seems interesting because they're sort of impeding on the coastal environment as well as sort of more of the bay environment.
Yeah.
So kind of both, and so it'd be interesting to see, just in terms of competition, how they compare to not only, crabs in the bay, but also, your coastal crabs, say the invasive Asian shore and green crabs.
Yeah.
Because I see them a lot just all together. And so it'd be interesting to see how they kind of impede on their invaded environment.
Sure. Yeah, that's a great point. Yep, absolutely.
All right, I have one question online from Sarah Davis. She's asking, will you be continuing to sample in upcoming years?
For the dredge survey specifically, or just in general? So maybe I should clarify. So these are all long-term monitoring surveys that are going on for a variety of different reasons, but they just happen to capture blue crab data. So all of these surveys will be ongoing in the future. The dredge survey will also be continuing, and then if it is a useful tool, we would continue it further than that. So yes.
All right, thanks very much, Katie. Okay, and then we have some remarks from Jeremy as well.
Right, well, I'll start by thanking our speakers for 4 very interesting presentations. My job is to provide some context for these talks to try to tie them together in the context of what we know about the benthos and Narragansett Bay. So I really have 2 observations about the talks. One is it's really gratifying to see that the sampling and experiments continued during the pandemic years, maybe not so much in 2020, but 21, 22. That's really gratifying that we've been able to keep getting out there and sampling. The second thing is that these sampling programs have really benefited by the comparisons that can be made with previous sampling. Some of our colleagues from URI who did this original sampling. I mean, it's one thing to go out and do a one year dredge survey, but if you don't have anything to compare it to, what can you say about, oh yeah, got some crabs. So being able to leverage, our current studies with historical data is really powerful. And we saw that in the benthic study with the juvenile lobsters, the studies that Merle has done, and also with the crab data. And so, we have been catching blue crabs actually since 1959. In the 1960s, somebody didn't write them down consistently, so that's why it's there. But also there's a long history of kids down on the little beach catching crabs with their pepperoni and hot dogs. I don't know if you can use that. It's been going on for a long time, probably including my kids. Okay, so I just have 3 slides. I don't want to keep you very long. We'll try to finish on time here, but provide a bit of context. The first slide is the coag or hard clam abundance from the DEM dredge survey. So these are data that were plotted by Patrick Barrett, who's the coag survey now. And he's provided time series for 3 regions. So you see the Providence River in red, the Narragansett Bay proper in green, and Greenwich Bay in blue. And these are the time series. So you see the sort of steady abundance of coags in Narragansett Bay. What's really interesting, what you notice, is uptick in the Providence River. That's suggesting something about the survival and growth of coags in Providence River as a mineral. So that's really, I think, ties into your study, Mike, to try to ask, what it is that's improved in the Providence River. So I think that's a nice link to make. Okay, go ahead. Okay. The second thing I did was look at the time series of benthic species in the GSO trawl survey to provide context for the other benthic studies. And so these are 32 benthic fish and vertebrates. And I thought that since Shane had already shown MDS plots, it would be okay for me to show another one. But it's basically showing the change in the community structure of these benthic species. This is for the GSO trawl survey grouped together from the 1950s when we started to the 2020s at the end. And you see there's a clear clustering of the community structure by decade moving across from right to left. And so you can interpret this change as basically this is time, or you can think of it as temperature increasing. And in the other axis, what happened in the 1990s was a real dominance of a number of invertebrate species. And just to show what some of the species are that are constituting these change, you see the big shift occurred in the mid-1980s between the 2 green clusters. And the next table here just shows what some of those species are that are accounting for that change. And the number of benthic eating fish, for many of them, you can see that higher abundance in the earlier group of years, the earlier stanza, relative to later stanza, winter flounder, ocean pout, sea stars, cunner, scup. Scup is actually a recent one, become more abundant. Hakes less abundant, cancer crabs a little more abundant. Now the toes tend to be dominated by things like little skates, and cancer crabs. Lobster actually was not very abundant in the early years. There was a peak in the 80s and 90s, and now, of course, as we heard from Declan, it's tailed off. But those are the species that are really accounting for the difference in the benthic community structure. And so we've gone from a cold water fish assemblage to a period that was dominated by invertebrates. Now we've gone back to a fish assemblage, but it's a different suite of fish species that are the benthic consumers. So this is important because these are the ones that are eating the benthos, so they're the ones where the benthic productivity is coming up to the higher profit levels. So I'll stop there and just say that this is ongoing work. Connor and I have plans to analyze these data in finer detail to drill down more. This is ongoing work. And I'll just end by saying that we really appreciate your comments, and I really encourage the students to follow up with the questionnaires to fully answer the question or get an input. I think it's very useful for your theses to do that. I've been taking some rough notes, so if you have any questions that you can contact me. And with that, I think we can wrap up right on time, 5:30, letting her go. So join me one more time in thanking our speakers from today. And thanks for coming.
Oh, yeah.
No.
Oh, really?